Okay, thank you, Audra. Sorry I don't have a bow tie, but welcome to the first full day, complete day of the conference. Before I get started, I'd like to say, although Ricky couldn't be here, I want to congratulate her again on putting together yet a wonderful weekend, and also congratulate Joanne for a much-deserved award last night. So there's really been some exciting things going on in research that I'm sure all of you want to hear about. Steve Rossi and Drs. McFarland and Trauner will be talking soon, but before they talk, I'd like to introduce Danny Williams, who's a sophomore in suburban Chicago, who's going to give a few minutes about PSC Partners. He's been involved for several years, so thank you very much, Danny.
Hi, everybody. Sorry I don't have a bow tie as well. Hi, everybody. I just want to thank Joanne and PSC Partners for giving me the opportunity to speak to you guys today. I'm Danny, and I was diagnosed with PSC at the age of 9, which means I've had it for around 6 years now. Now being 15 and a teenager with PSC, I view it differently than I ever have before. I try to use it as an advantage or an opportunity to grow as a person, socially and mentally. The first time I went to the conference was last year, where I met so many new faces and some familiar ones. Having that experience of coming here as a teen and being a part of our group has been very memorable to me. It's impacting me in ways I've never thought before. Whether it's sitting around on some beanbags, talking about life stories, or hanging out playing board games, or getting up and going to explore. We do it all. As a group, we individually have our own differences and backgrounds, but when we're together, we're all united. I also want to talk about Zoom rooms. Zoom rooms are very helpful for us to stay connected and keep up with each other's goals or personal stories. It's a great way to stay in touch with our teen group if you're unable to make it to these conferences. In the end, being a team with PSC may be uncomfortable and discouraging, but everyone is here for each other. I also want to thank the doctors and specialists that are here supporting us teens. And remember, no one's ever alone. Thank you.
Thank you, Danny. I think that really puts a perspective on the rest of the day and why we're all here and our common goals, which hopefully we're getting closer to every day. So I'd like to introduce our first speaker, who is Steve Rossi, who I think many or most of you know is the Chief Scientific Officer of PSC Partners. He's worked with chronic liver disease patients for over 30 years, both as a clinician and researcher. For the past 20 years, he's been working on drug development, and he brings a wealth of experience in clinical research to help lead PSC Partners, including the WIN-PSC study and the International Collaborative Network of PSC Partners Research. So thank you very much, and I'd like to introduce Steve.
Thank you. Okay. Well, thank you all. And you guys hear it? There we go. It's great to see everybody here again this year. And over the past 2 days, or yesterday and today, you've heard so much, you're going to hear and have seen so much good research being done by many of the people in the room. But I think especially for the newcomers, many of you don't realize that PSC Partners itself is a very strong research force. And I'm going to talk about one of the aspects that's been sort of the legacy of PSC Partners in terms of supporting research, and that is our research grants program, which has been awarded to many people in the room and has really moved some unique projects forward. Now, several years ago, we did get a grant from Chan Zuckerberg, and that really allowed us to launch and decide what was the research strategy that was really going to be part of how we move different projects forward. And over 400 of the community members participated to really help us identify what was a priority for you. So the scientists have all kinds of amazing ideas in how to move the needle forward, but ultimately, PSC Partners wants to do research and support research that really reflects what you see as important and really helping to better understand the disease. So there were 4 big areas that came out when we took in all these research ideas. And again, we did focus groups, we did all kinds of ways to network with lots of patients and scientists. The first was really better understanding PSC as a disease and how it progresses and why. The second, and we had a great meeting the day before this, and you saw from Dr. Korzenick's presentation, what is that relationship between PSC and IBD? The third, and one of the most important, and we saw again that there's a lot of advancements being made, and hopefully in the near future we will see some drugs that can hopefully either halt or reverse the disease, and importantly also work with symptoms. And then lastly, it's a tough area to focus on, but understanding and really helping us better detect cholangiocarcinoma and educate everybody around the risks and also the opportunities for treatment or different aspects of cholangiocarcinoma. So all of our research that we do now fits into one of these 4 buckets. And it really started with your voice that helped us kind of decide how we'd move forward. Now, the PSC Partners research team is really focusing on a wide range of projects, and I encourage you to go out to the research booth and you can learn more about these. But we have a variety of big programs, everything from areas that we're supporting to where we're actually leading the research. One of our biggest projects is the WIN-PSC, which is a global prospective cohort that's being conducted at up to 20 sites around the world to really better understand how PSC progresses and also serve as a data set that can potentially support new drugs being developed. We have our International Collaborative Research Network, which brings together patients and experts in the community to define new projects. And you'll hear more about one of those projects later today from Dr. Pedro Rodriguez, really trying to build patient-driven and partnered research in some of our areas. The Symptom Assessment Project that Donna Yvonne is leading, developing tools for fatigue, brain fog, and liver pain that can be used in trials, and that's really moving nicely. The registry, which Brian Thorson is leading, is also part of our research program, and there's opportunities there. This last couple years, we launched 2 big surveys in conjunction with the registry and also PSC support over in the UK. The acute cholangitis survey, many of you may have participated in, really focused on what are the symptoms and the patient experience in this very, potentially life-threatening condition. And then we recently launched the cholangiocarcinoma survey. Both of those, we got 600 patients who actually participated. And we really encourage you to participate in those because they really do give us information. And in fact, the CCA survey is actually being presented at the AASLD liver meeting coming up in the fall. But our grants program is sort of the foundation of where research and the partnership with research and PSC partners really started to make connections with the community. And this has been around since 2007. Rich was actually very instrumental in continuing to help drive that program forward over the years. And we really have 2 different types of grants that we really focus on. The first is what we call our standard investigator grants, and these are people who are established researchers that really are bringing forward either new or advancing ideas, and we support them with up to $75,000 over a 2-year period. But something that we've now made a real commitment to is helping young investigators, who are the next generation of researchers and treaters, and how can we actually help support them and launch their careers. And so we give a little bit more support to those grants, but they're so important in helping build the next generation of experts who will really help, I think, move some of those research areas forward. Now, we sponsor everything from preclinical research to actually clinical trials. We're seeing more and more sort of novel approaches of really unique, very high caliber research. And when the program started, we'd get maybe 8 to 10 applications that would come in. I think this last year we had almost 40 applications that came in of high caliber, very, very, very competitive research programs, and we're able to fund some of those, as you'll see. Now, the impact of these grants is actually broader than just PSC Partners in the US. And as you can see, over the years, We funded over 100 programs and grants. More recently, every year now, we do fund 2 young investigator grants, and those will continue to be a major force behind what we do. PSC Partners Canada does partner with our program and actually funds a grant themselves. And really, those can be anywhere around the world. And they have sponsored some really unique programs in conjunction with us. And our reach has actually spread from not only the US and Canada, but we actually have funded quite a number of grants for some of the top researchers in Europe as well. Now, the program did start quite small, and as I said, it first started funding grants of a smaller amount back in 2007, but those were really an important start for where things actually were able to provide some financial support for good research. And it is so critical to remember, this is where many of your donations and dollars go to actually, if you want to see where your money, when you give to PSC Partners, makes an impact, this is one of the big places it does, and it continues to grow in our ability to actually sponsor research. A few years later, we hit actually a great milestone, and that continued for a period of time. But you can see, most recently, up to this year, we've now sponsored over $5.4 million in grants to drive research forward to find that cure for PSC. Now, it's not only made an impact to the field, but we've been very lucky, and some of the people who received the grants and who are on this slide are actually in the room today. A big impact that we've seen is through some of these seed grants, different people have expressed how these grants have helped them either launch their careers, build new collaborations, or actually provide data that allows them to then proceed and get NIH grants, different career development grants. So the program really does lead to building, again, a broader community and really helping sponsor more research to continue. Dr. Lee has worked with us from UCSF and has built some nice collaborations, really focusing not only on sort of the detection of what might be antibodies associated with PSC, but also working with us on some of our symptom network. Dr. O'Hara at Mayo has actually received 2 grants and has really been expressed so strongly how our grants have helped him pursue other grant opportunities. And Dr. Popov actually was very strong in supporting us and acknowledging how we helped him support the development of some new models to look at fibrosis. These continue, and what I'll encourage you is we do have an impact report at the booth, and it really can give you a good sense of what can be done and what we have done with the research grants. Now, our current grantees this year, we're very happy, and again, several of them are in the room. Our 2 standard grants were Dr. Zhang and Dr. Awani, both of them looking at unique pathogenesis aspects of PSC and different cell types that might be important in progressing with fibrosis. And Dr. McParland, I apologize, you were actually the recipient of our other standard award, and I think what you'll see with Dr. McParland next is how she's working in the basic science field, but so importantly, bringing the patient into that partnership. And then, as I mentioned, Dr. Awani and Drs. Hoyle were our young investigators this year, and large influence of basic science work, but we also do get some very important clinical grants. And as you can see, Dr. Swain is actually focusing on some, what I call the science of symptoms, and really focusing in on something that's so impactful for the patients, and that being fatigue, and how to really better understand that. Now, where does the patient actually become a part of actually the process here? We actually evolved over the last few years to have the patients actually have an equal say in what grants are selected. So we have a very strong scientific medical advisory committee that Rich has led for years with Dr. Chris Bolus. It's going to be led this next year with Drs. Popov and Drs. Bolus leading us forward. But the community has its own say in this, and we have a very structured way that we actually look at the grants. There are different ways that we look at how patients can be participating in research. Everything from being sort of a consumer of different aspects of things that are being developed, all the way up to like what we're doing with wind, where we as patients are leading and driving the research and collaborating directly with the researchers. We take a lot of that and we give strong credence to projects that actually involve the patients at more in the patient-partnered area. But we also review the grants with a structured group of reviewers that look at how does it align with our corporate organizational mission. Is it a priority based on what you guys have said is a priority for us to better understand? How does it fit to other things being done in the field? And really, will it make a big, how broad will that impact be in the community? Now, for many of you, if you look for opportunities to how to be involved, we actually do have and are looking for additional community reviewers. So please come out to the booth if you're interested in being part of the process and actually not just providing your voice on what we do, but actually being a part of the process. Now, our grant cycle did just launch, and I shouldn't be up here. It's actually Sarah Kurab-Khalif who actually is driving this program and is part of the research team I lead. And she has taken us to another level from basically a very sort of more grassroots approach to everything being online, automated, really being able to handle 40 applications that we probably wouldn't have been able to do in the past. But we've started the cycle with the letters of intent, and we've already gotten our first letters of intent, and actually some people are working on their applications. And at this stage, we are now actually requiring the grantees to have a patient partnership plan as part of their projects, even if it's a basic science project. And many people say, how does that work out? Dr. McParland, I think, is going to really give us some insights into that. We'll then do the reviewing, scoring, we'll award the grants, and then year one funding commences. Generally in about the May timeframe is when we'll see the next wave of grants come through. And then we finish 2 years later with actually a final report. And what we will start doing at the conference, and actually with some of our education, is start to bring the results of that research out to you so you hear and see the things that you've supported over the years. So I mentioned a few areas and opportunities of how you can actually be involved, and so many of you have already given your time, your thoughts. I can give, for an example, our ICRN project, the cholangiocarcinoma project. We had 6 high-quality proposals that were put out there from the ICRN, and we pitched them out to the community, and you guys chose the one that mattered, and this was in the area of cholangiocarcinoma. So when we ask for your feedback, just know it does make a difference. But there are other areas where you can be involved as a reviewer. You can become a patient partner if interested on some of the basic science research, and obviously donating and supporting to the programs. But we can't thank you all enough as the community members, our SMAC, who's helped drive sort of making sure we're doing scientifically rigorous work, and all the team that works to bring our research grants program forward. So thank you very much from our team, and we're excited to see new programs and new research drive forward. So, yeah.
So thank you, Steve. We're going to have a panel discussion at the end where we'll be able to take questions, and I think actually there's going to be cards passed out if you want to write them down. Our next speaker is Dr. Sonia McFarland, who's a senior scientist at the Ajmer Transplant Center at UHN and an associate professor at the University of Toronto. She's in the Department of Laboratory Medicine and Pathobiology and Department of Immunology. And her research programs focus on the translating fundamental knowledge of the immunology of the liver into clinical applications. And then she'll talk about there's been some incredible technical advances in single cell ways to look at livers and other organs, and her group has really been at the forefront of defining this in many diseases, and PSC in particular. Her team developed the first transcriptomic gene expression map of the human liver, and this has helped explain the immune dysregulation of liver disease, as she'll be talking about. So I'd like to welcome Dr. Sonia McFarland.
Thanks. Thank you so much, and thank you to Danny for your compelling story. So today I'm going to talk about patient partnership in research, and I will just touch on my research very lightly in just really introducing how I got into PSC research and how our team got into PSC research. And so, as Rich mentioned, we had worked prior to being in PSC research in mapping the cells that make up the liver as a platform in which to understand how livers become diseased and how we can push them back to a healthy liver. So once we had generated that map, we spoke to Aaliyah and Gideon in the hepatology group, and we said, look, if you could now direct us to the first disease you want us to describe, what would it be? And they said, PSC patients worry us the most. And they focused us on what their biggest clinical concern is. And that's how we got into PSC research. And what has been very important and very special about this project is that throughout the course of this project, we've really been able to do this with patient partners and with the patients and a focus. And what I'm going to talk about today is how having patients on the research team makes the research team better, and it also speeds the research forward. So I will start, first of all, just defining what is patient-partnered research. So it's about engaging patients and their families as partners in the research process to make sure that the studies are on the right track. They're patient-identified priorities, and when we come to meetings like this and we present, the patients say, this is a question I think needed to be answered. And so the other part of patient-partnered research is it's important to learn from patients. So it's important not to just sit there and deliver the research program to the patients, it's to learn from patients. And so I'll give you an example. As we wrote our grant for the first grant that we applied for PSC, we approached the patients and said, if you were to give a biopsy, would you rather give a biopsy on the back bench for a shorter period of time, or would you rather go to interventional radiology, spend a longer period of time but potentially be a bit more comfortable? And they said, absolutely, we would like to do it through IGT, be a bit more comfortable, and that will actually make the process much better for us. And we incorporated that into our protocol. So there's backwards and forwards learning, and it's a very kind of genuine relationship. And so it's important to picture research as doing things with people rather than doing things for people or to people. And the important thing that happens when you do this is the patients participating in the research, it grounds you in the why. And an interesting question came up last night from Andy Dench, if he's here, and he said, research is slow. And I'm like, yeah, it's pretty slow. He's like, how do you stay motivated? And one of the ways to stay motivated, in fact, is to have patients in the room. Because when you go to funding agencies, they say, well, where's your Nature paper from last year? Where's your Science paper from last year? But when patients are in the room and they're hearing the progress and they're seeing the students presenting the challenges and also the successes, they actually can say, you guys are really making a good effort. And it really grounds you. And they say, this makes it, easier for me to understand how my disease is potentially progressing, and it makes me feel hopeful for the future of research in PSC. So it grounds you in the why on days where experiments are not going so well. And so, but these relationships can be tokenistic if they're not following specific principles. So to have a genuine patient-partnered relationship, you have to be inclusive. So you have to make sure that patients' perspectives are actually being included. You have to make sure that there's support. So you cannot have a research meeting that spans 8 hours straight with no break and expect PSC patient partners or their families to sit through this whole thing. You have to make sure that you're thoughtfully planning the collaboration and the partnership with the patients in mind. There has to be mutual respect. So you have to always kind of finish a meeting with, from the patient perspective, what could we be doing better? Are we wasting your time? And then often with our patient partners, it comes back to, are we wasting your time asking these questions? And the answer is always inevitably no, but that communication really makes everyone feel very at ease with their role in the research project. And then co-building is very important. So one of the things that was important to us as we learned about having patients in our partnership is not saying, here's the research program that we wrote, here's the grant we wrote. Now we saw from the funders that we need a patient partner. We're going to bring you in just before we submit the grant. So that is not co-building, because then as they look through the grant, it's like, well, this is not a question that we think is important. And so it's important to engage patients thoughtfully right from the beginning. And so that is what we have been trying to do. And so really the linchpin of our patient partnership is Mary Vyas. A lot of people in the room would know Mary. She's the parent of a young adult with PSC, and she's the co-founder and president of PSC Partners Canada. And a lot of people, when we talk about our patient partnership, say, how did you guys meet? And so the way that we met, in fact, was that Mary did exactly what you're all doing right now. Came to conferences, listened to the scientific talks, listened to the basic as well as the clinical talks, and came up after and engaged with the speakers. And we were at a conference in Montreal, and Mary found us and really started talking to us about our research, and then we organically became a part of a team. And so she started, this was in 2019, so we were applying for a grant from the Canadian Institutes of Health Research. So we incorporated Mary as part of our team, and we didn't really know how valuable it would be until we started to see how our team actually grew through her participation. So she brought people together. So taking the time to come to meetings, to go to talks, to engage with the speakers after, it is so valuable. And this is, and I'll talk about all the kind of the value that Mary brings to the table, but this actually establishing the collaboration, that was all on Mary Vyas and Rachel Gomel. And so in that first kind of collaboration, that first grant, Mary kind of decided then to bring other people in, including Rachel Gomel, Jessica Travis, and Verva Alges. And within our University of Toronto team, we had myself, we had Gary Bader, who some of you would have heard talk about our mapping efforts, Amanda Ricciuto, and Diana. So this was for the pediatric part of the grant. And so within that collaboration, we started to study the cellular microenvironment of the pediatric liver. And then, and then as the collaboration developed, and we started to see that we can use these maps to identify potential targets that we could test, then Mary identified that we needed to actually bring in Steve Rossi to really understand that part of the scientific process that basic scientists would never know. So it makes it more efficient when you have someone, a patient identifying, you know what, this person needs to be in the room because this is where they're eventually going to have to take over, and they're going to have to bring this to a therapy. And so this was a really key value of Mary. And then within this program, Mary and her team were always presenting to patients and keeping patients kind of up to date with what we were doing. And so what were the specific activities of the patients? So the patient group was organizing and hosting conferences, recruiting patients to the registry, sharing resources, and welcoming collaboration. And there was a concrete contribution because as Mary got involved and as the patients then were going to get in and actually participating in research-only biopsies. So one of the things that really came up on Thursday is we're starting to understand PSC at the time of transplant, but we really want to treat early. And if you want to have early stage biopsies, a lot of these need to be done through research-only biopsies. And so this is the power of the patient group. The patients understood the project, they bought into the project, get in, explain the project, and Mary kind of also helped with that. And then internationally, we're a unique group in that we can look at research-only biopsies. And sorry, from the research group perspective, in order to really show that we're also giving the data back to the patients and making it so that this is your data and your research, we've generated tools that can share the data. So no one has to come to us for the data. They can go to Mary for the data. We share them openly online. Anyone can have the data so that we're pushing together forward science for every group. And so we made sure that all our protocols are online. We make sure that all of our data is publicly available. we don't hide anything until publication, because we understand that this is not about us, this is about the science and about the disease and about information. And then we have joint activities. So Mary and Rachel will participate in our monthly research meetings. And so these meetings are not very formal. There's the Gideon and Jordan and Aaliyah and the basic team and the patients. We're all there and we're just talking through data. Half the time we're talking through data that doesn't make sense, or we're talking through challenges, but having the patients in the room, we really do, it does raise different questions, and really what we make sure we do is we check in and make sure, like, did this waste your time? And often Mary says, and Rachel say, this is actually really fun for me, and if you're wasting my time, I'll let you know. And then Mary and her group really anticipate challenges. Okay, we say we want to recruit patients. Then you need to take your fancy science and you need to put it in the right language for patients that are maybe not English speaking, so they work on translating, and also just making it accessible to people that might not be working in a lab 24/7. And so with that in mind, we've really launched 2 very cool projects. Number one, characterizing PSC at different stages of progression, and that is something very unique to this group, and it's only because patients are going to Gideon and saying, we can do research, we'd like to give a research-only biopsy. And this will help the field in general, so we can understand targets that exist early in disease that may be separate from those kind of more cirrhotic, more end-stage signals. And so this is a collaboration with PSC partners, as well as with Sick Kids, with Toronto General, including the hepatology and the transplant program. And then the second one is a CZI patient-partnered collaboration. And again, this was a really interesting collaboration because a patient advocate or a patient member needed to be part of the writing process and needed to be actually a co-applicant. So patients, the patient group and Mary and her team actually had a budget from this, and I think that this is the way that funders need to proceed. If they really want to have genuine patient partnership, you need to pay for the patient's time, and you need to have a mechanism to pay for meetings and really bring patients together. And this is what was done through the Chan Zuckerberg Initiative. And through that, we're looking at the gut and the liver in PSC patients and to see shared disease signals between IBD and PSC. And so, and lastly, I really want to just go through how this looks beyond being in the lab. So twice a year, 3 times a year, and Matt really highlighted it last night, we have road shows where we talk about the research. And people ask really fascinating questions. And actually, it was really interesting because the first time we presented, when it was question period, it was crickets. And so Mary very gently said, I think you presented maybe too high level. Maybe you need to really present in a way that's more accessible. And so what she did was she had Verva, who is a patient communicator, go through our slides and say, this may be very meaningful for you, this makes no sense to the patients. So change that, explain it at a way that's accessible. And when Verva had gone through our slides and basically told us what to rewrite and make it more accessible, there was questions all around the room. So we're really starting to learn how to communicate our projects, and that is actually leading to more participation from patients. And people are emailing saying, can I send you a biopsy? I'm in Alberta. And that's really cool. So that's exactly what we're trying to accomplish. And this actually led to us having the Trans Zuckerberg funds leveraged through the Conscience Network to increase those funds and really to bring us some mentorship into how to really take a repurposed drug and to apply that in an open science type of way. So it's really expanded our research program, and that's really due to the participation of the PSC Partners team. And then the other cool thing is that we had patients kind of say, you know what would be cool? We'd like to see our diseased liver like in 3D. We want to see what those strictures look like. And so Albert Fung at the Toronto General made this app that he brought last year to Phoenix, and really that has helped us really explain the disease in a better way. And of course, we enjoy these kind of these organized knowledge translation events. And so it really does lead to unique questions that we actually apply in our grants. And the last example I will have is our PSC-funded project. So last year when we were in Phoenix, a lot of people, we kind of said, what would you do next if you were us? And a lot of people said, what does the common bile duct look like? What does my stricture look like? What cells make up that diseased area? And so we went to the surgeons and said, this is what the patients want to see. What can you do for us? And the surgeon said, we can take you off some healthy bile duct, common bile duct, and we can take some diseased common bile duct, and you can profile them. And so we're actually doing that now with the funding of the PSC partners, and this is the most, and this is something that's really being led by Diana Nakib, who just recently got her PhD. So this is something that will absolutely be a patient-led. It was a patient's idea. We do it, and we'll give the data right back to you. So this is a very genuine example of a patient-led project. And so in conclusion, I just want to thank everybody, and there's definitely people that I've left off this slide, but it really does take a huge team, and people's time is very valuable. So one of the things, I just really want to thank the patient partners. You really do enrich our lives. You make us grounded in the why, and you really do motivate us when there are days where the research is just not really working out and we don't really understand why. Thank you.
Thank you, Sonia, for that excellent talk and putting in perspective of what you do and how basic science can be very patient-oriented. Our last speaker is Dr. Michael Trauner, who's from the Medical University of Vienna. He's a professor of medicine there. He originally trained at the Karl Franzen University in Graz, did postdoctoral research, including at Yale with, as we found out yesterday, Chris Bolus, as well as Josh Kurzinik, and has been a leading, one of the premier international investigators in basic and clinical science in the field of cholestatic liver disease. He's going to talk today about what may be one of the fundamental clinical advances all of us have been waiting for, so it's my esteemed pleasure to introduce Michael Trauner.
Thank you very much. Thank you very much, Rich. Thanks a lot for inviting me. I really love being here. I think this is the 5th PSC Partners meeting for me, but I think I never talked to the entire group. So it's a really pleasure to present some of the recent progress. I was thinking about, what should I really focus on, since there are so many exciting sessions about progress in PSC. So I thought I would focus on the lack of medical therapies. As you are well aware, in contrast to PBC, the little sister of PSC, we have no established drug treatment, right? And we'll focus on that, and also the link to IBD. I won't focus so much on malignancy risk in my presentation. So one of the real challenges and uncertainties for you as patients is the course of the disease, right? It's so difficult to predict. Biochemical lab parameters fluctuate, the symptoms fluctuate, IBD activity is fluctuating. So the only thing which is giving you kind of a good picture of what may be going on is liver fibrosis. And this is a common theme to virtually any liver disease. And the diagram here on the right-hand side is showing you the progression of liver fibrosis over different stages. So F4 is liver cirrhosis, which is the maximum. F0 means no liver fibrosis. And for you as patients, of course, it's very important to know, where do you stand? You see in the left part of this curve, the curve is pretty flat, right? And many of you stay there forever in this flat part and may never progress. The problem is it's difficult to predict who is going to progress. So the only way is to monitor liver fibrosis. And also, as you see in this diagram, this is not a linear progression over time, but you suddenly get an exponential increase, which is in this space of what we call F2, F3 fibrosis, which means significant or advanced fibrosis, which then has a high risk of progressing to liver cirrhosis. And this also means that for clinical trials, for example, it is very important where you're looking at. In some clinical trials, when you look at patients who are in the stable part, nothing may happen forever. It takes a very long time, much longer than the clinical trial lasts, that you will observe any changes. So also for clinical trials, it's very important to focus where this curve is actually changing. And this is something which is also not unique to PSC. This is also what is done, for example, in steatotic liver disease, that when we have significant or advanced fibrosis and we can diagnose that non-invasively with FibroScan or serum parameters like ELF, this is actually where the time for action is. And this is also something to keep in mind, because liver fibrosis is driving clinical events. So progression to liver cirrhosis, decompensation, ultimately liver transplantation, preventing death. But you see that, for example, malignancy and cholangitis, which of course are extremely important clinical events in PSC, do not follow this trajectory of liver fibrosis. They may occur a little bit earlier and independent of liver fibrosis. And this is the thing which we are struggling with when we design clinical trials, who of course integrate all the totality of data, including cholangitis, which of course matters especially to symptoms such as fatigue, for example. So we now also in our clinical practice guidelines actually try to classify or determine where is there a significant risk. And this is basically, especially at this turning point where liver stiffness is in this space where the curve becomes steeper. So basically liver stiffness or 10 or an ELF score of 10 is telling you this is something where something may happen in the next time. So let's look a little bit closer. And also in clinical management now with FibroScan, that was confirmed by the FICUS trial, a European FibroScan trial presented a few years ago. And also in our clinical guidelines, actually, we try to determine is there low risk or is there a higher risk. And when there's a higher risk at this rate where the fibrosis progression curve may change, this means one has to have a little bit closer looks. Once a year may not be enough. Perhaps this means you have to visit your doctor every 3 months or every half year. And of course, in addition to these non-invasive fibrosis parameters such as FibroScan or ELF score, also imaging plays a very important role, not only for cancer surveillance, but also MR imaging is also giving you prognostic information. There's the Annales score, what the liver is, in which shape the liver is. And of course, endoscopy, meaning colonoscopy, for those of you with IBD, is very important for surveillance of colorectal cancer risk. So let's have a look at the pillars of PSC therapy. And I owe this slide to Michael Manns, who is really, he was one of the big shots in hepatology, who really dedicated a lot of his life in PSC research. Unfortunately, he passed away tragically and unexpectedly 2 weeks actually ago. And he kind of determined in this very educative slide, there are 3 pillars of PSC therapy, right? There's ERSO, there's endoscopy, and there's liver transplantation. And as you see, this column of ERSO has a crack, and this crack comes from the uncertainty whether ERSO actually is really going to benefit, whether there is an extension of lifespan. And also, as you are all aware, there was this high-dose trial from the Mayo Clinic by Keith Linder, which showed that even higher doses may even be detrimental. And I want to show this slide about endoscopy, about ERCP, before I turn towards pharmacotherapy. We are all very restrictive about ERCP. Many of us, physicians, of course patients, don't like ERCP. But I want to highlight those 2 studies, which I think we have to reflect on those data. We cannot ignore those data. ERCP is uncomfortable, it is risky, but maybe we can make it safer, because these studies show that patients who actually are recalled on a regular scheme, like for example for colonoscopy, you do the same thing for ERCP, have a much better survival, which is the other upper curve, than when you wait for a clinical event, for example, like deterioration of liver enzymes or a cholangitis. And also you have seen this image several times at this conference already, which is an advanced way to look into the bile duct, which is cholangioscopy, where you see the inflammation of the bile duct. So really the bile duct is at the center of the disease, and also Sonia's comment on the extrahepatic bile duct actually, I think, highlighted that. I think we have to think about making the procedure more comfortable and safer, and not just avoid it because it's so complicated to train people, to train physicians doing this procedure safely. So I think it's a technical problem which can be solved. And also when it comes to PSC, I think the topic of mucosal healing is extremely important, like it is, for example, in IBD. We don't know at all what is happening to the bile duct when we treat PSC with the drug. Is the bile duct healing? You know, is the mucosa healing? I think those are questions which really need to be determined in the future. So let's have a look what kind of drugs are available at the moment. And this is a very overloaded slide with a lot of information, with a lot of drugs which are currently tested in PSC. And basically, you can look at it as 3 groups. They are bile acid-based therapies, or the PPARs, the nuclear hormone receptors like elafibranor, which you've heard about, or urso and norurso, which I will talk about. There are microbiota-based therapies such as vancomycin or fecal microbiota transplantation. And all of those approaches actually have shown a positive ALP signal, which is this check signal at the right side. Intriguingly, like in PBC, also in PSC, immune modulatory therapies or therapies for IBD actually are not effective. And we really don't understand that. I think for PSC, one explanation may be that we just simply diagnose the disease too late. I think we have to get rid of the sclerosing in the terminology of PSC, like we got rid of the cirrhosis in the terminology of PBC. We just simply have to diagnose the disease earlier with, there are imaging advances with MR. There's even PET imaging, which now has become with lower radiation exposure with the modern PET machines. And perhaps we even have to do cholangioscopy, with safe little thin catheters which can be inserted to look into the bile duct, whether it is inflamed before we have the stricture. The stricture is basically the sign of an end-stage disease, and Michael Manns, whom I mentioned you earlier, always referred to PSC as the cirrhosis of the bile duct. Who would diagnose hepatitis C based on the criteria that there has to be liver cirrhosis that you make the diagnosis? So I think it's counterintuitive, and we have to move the field forward to diagnose PSC at this stage when there's cholangitis and not sclerosing stricture, and that may require a redefinition of the disease. And which other treatments do we have? So I want to focus on norocolic acid, or norurso as it was called before. So this is basically a derivative of ursodeoxycholic acid. So it's shortened by one methyl group by this little box. So a very small modification is changing the molecule. And this was an academic discovery. So this is also a very important take-home message for you, that norocolic acid was discovered and developed in academia and then was taken over by industry and developed as a drug. So it's really a prime example of academic industry partnership and how that can go well. And actually, Alan Hoffman developed this compound to demonstrate the process of colohepatic shunting, which means a shortcut between the bile duct epithelium and the liver, which results in bicarbonate secretion. And this is shown on this slide. So a very important way of the bile duct to protect itself against bile acids is to produce bicarbonate. And by undergoing this shortcut, this colohepatic shunting between the bile duct and between the hepatocyte, you induce bicarbonate secretion. And also that was the concept when we moved this molecule into preclinical testing in a mouse model of sclerosing cholangitis, which was is shown here, which is developing, these cholangiographic features of PSC and also the typical histological changes. And these could be totally reverted by NO-UDCA in the preclinical setting. And the mechanism behind that is the targeting to the diseased bile duct through this colohepatic shunting. So this bile acid is not undergoing the circle through the gut, the so-called enterohepatic cycling, but is undergoing a shortcut, this colohepatic cycle, and also has anti-inflammatory and anti-proliferative effects. And actually, we were very much interested whether that also could be used to treat inflammatory bowel disease, because both in IBD and PSC, we have pretty much the same changes ongoing with the disbalance between the good immune cells, the regulatory T cells, the Tregs, and the bad immune cells which cause inflammation, the Th17 cells. And we tested this in mouse models of gut inflammation, and I'm showing you just one histology. If you focus on the right hand, this is the mouse which was treated with NO-UDCA, where colitis was totally reversed, the architecture was normalized, there are goblet cells producing the mucin barrier, and there is a non-inflamed gut integrity. Without treatment, you have this severe inflammation. And actually the mechanism behind that is, and you've heard that yesterday in the industry presentation, is actually metabolic rewiring of the immune cells through neurocholic acid. So this Krebs cycle, this energy cycle, which was mentioned yesterday, going on between the mitochondria and the cytoplasm, is actually rewired. So this is a big topic at the moment, that you change inflammation by changing metabolism. And this dual impact on liver injury and colitis in mouse models, of course, is very attractive to treat PSC. And this led to the clinical development of neurocholic acid from preclinical mouse models to phase 2, phase 3 studies. And you see, this is the typical funnel of clinical development. Usually only 1 out of 30 or 40,000 compounds will actually make it to a positive phase 3 study, which happened with neurocholic acid. You've seen some of the data already yesterday presented by Gideon, and he presented to you also the study design. So it's a 4-year study where the interim data now, the 2-year data have been presented and analyzed. And the combined endpoint in this trial is the improvement of alkaline phosphatase and the stabilization of liver histology based on the Ludwig stage, which is mainly reflecting liver fibrosis, which I explained you is determining the progression of the disease. And this endpoint was hit in 15% under neurocholic acid versus 4% in placebo, which doesn't sound very much, but you have to keep in mind that study was performed during COVID, during the Ukraine-Russian war, and during Brexit. So many patients just simply didn't come for the second liver biopsy and had to be counted as treatment failure. When you focus on the patients who only had a liver biopsy, which is called per protocol analysis, you actually have a 20%, 22% chance, 1 in 5, to respond. But more interestingly are actually the histological changes, which Gideon didn't show you yesterday. So shown here is actually whether patients improved liver fibrosis, shown here in the upper light blue box, whether they deteriorated liver fibrosis in the dark blue, or in the middle, whether liver fibrosis remained the same. When you look at the progression of liver fibrosis, you see that under placebo, actually 40% progressed and only 20% under NCA. So this was the same endpoint like in the PRIMIS study, which you've heard about, the Gilead study with Silofexor, which was negative. So NCA actually slowed down the progression. And perhaps more importantly, it actually it increased the improvement of fibrosis, which is the light blue, the upper part. 25% actually improved under NCA versus 11% under placebo. And importantly, also the progression to liver cirrhosis was basically cut to half, which is a very important clinical surrogate parameter. And what very much surprised us when we looked at the patients who did not receive ursodeoxycholic acid, the response to urso, shown on the very left with the 31%, was much higher and was actually weakened by ursodeoxycholic acid, which itself did not have an impact on liver histology. So I think this may be another crack in the question whether urso is really useful in the treatment of PSC. And there's another way of looking at that. It looks complicated at first glance, but actually when you look at the patient who deteriorated liver fibrosis in the different stages, and you see most of the patients actually were in this stage 2, stage 3 category of advanced significant fibrosis, and you see on the left under NSAA there were less patients progressing to advanced fibrosis than under placebo, where actually twice as many progressed. And on the other hand, importantly, also the improvement of fibrosis was twofold higher under NSAA than compared to placebo. So patients improved by one fibrosis stage from stage 3 to stage 2 in 40%. Some even went back to stage 1. So for the first time, we also see improvement of liver fibrosis, which was a very surprising but positive signal for all of us. And these liver biopsies are not only very important as a surrogate parameter for clinical outcomes, which of course in 2 years cannot be monitored, but also for research. And what Sonia has explained to you, having now liver biopsies before and after drug treatment and the baseline in 300 patients is an enormous repository for research where we really can look into the mechanisms of PSC and how actually a drug who now has been shown to work may have its mechanism. And we are very grateful to the patients who underwent liver biopsy in a condition where liver biopsy is not part of a clinical routine. So apart from research dedicated biopsies like Sonia has explained, having liver biopsies in clinical trials is a very important resource, not only as a readout, but also to understand the disease and the mechanism of drugs and how this can be further enhanced. So basically, we hope, of course, that this will result in approval by EMA. There's a grain of salt in this study. There was unfortunately no impact on patient-reported outcomes like fatigue and pruritus. You also see that there's an extremely wide distribution. Those bars, stretch over a wide region. So the symptom severity was very variable. So we are now looking more carefully, maybe patients who had more severe fatigue or pruritus whether they improved. And as you heard, very importantly, the drug was also very safe. So the journey is not yet over. We certainly have further avenues to look at. I think that elafibranor is an extremely exciting compound. You've also heard about the data yesterday. Probably we have to combine drugs in a complicated disease like in PSC. And especially the fibrates may have the benefit and the PPAs that they have been shown in other diseases to have a positive impact on pruritus. And when we think about microbiota-based therapy, let's not forget that diet is the most powerful or one of the very powerful ways of modifying gut microbiota and modifying also the metabolism of microbiota, for example, of bile acids, which then could have a beneficial impact. So the pipeline, of drug treatments is really full, luckily. So I've shown you the data about norocolic acid. I've mentioned that unfortunately the cilofexor trial, the FXR agonist, was stopped. It was a negative trial. There is a bezafibrate trial ongoing, a phase 3 trial in Europe at the moment, led by the Paris group. And hopefully we will see phase 3 trials with elafibranor or some of the other antifibrotic immunomodulatory drugs. So with this, I want to close. PSC remains a very heterogeneous condition. It's a very individual course of the disease, which of course is causing a lot of uncertainty to you as patients and also for us as doctors. The only way is to monitor non-invasively with non-invasive fibrosis tests, and we have now several promising therapeutic strategies, with norocolic acid being the first positive phase 3 study in PSC. We'll see what microbiome and immunomodulatory treatments will show in the future. I think that early diagnosis is essential to make those treatments more effective, and in a complicated, complex disease like PSC, we will have to combine different drugs in the future, like we do it in diabetes or arterial hypertension. And of course, patient-related outcomes is a huge unmet need, and we still lack effective therapies which are combating fatigue. We have made some progress in pruritus. I think this is also a major research direction. So with this, I want to close, and I hope I still left some time for discussion. Thank you very much for attention.
So I think, and Audrey, you can correct me, we have till 10:45 for questions, is that correct? So we have about a half hour where we'll be taking questions. Please write them on your cards, and we have a distinguished panel, and I will ask the first question. The COVID vaccine came very quickly, and it was based on roughly 10 or more years of research that probably few, if anybody, in this room knew about. So we all, I think, understand when there's a phase 1 or 2 or 3 clinical trial, how we take a putative drug and figure out how to dose it, and hopefully eventually it'll come to drug approval. But perhaps you three can comment on, what happened the 10 years prior to that in COVID relative to PSC. Why should we be doing liver biopsy studies or animal studies or ERCP studies in the hope that we could get to a cure or a very effective treatment 5-10 years later?
I guess I have to take that question first. The mic is not yet on. Okay, do we have handheld? Yeah, now it's on. I think it's the complexity of the disease at the end. Also, in order to explain the drug development progress, when we went to EMA to discuss the study design, the European Drug Agency, we didn't want to do liver biopsy. The drug agency and also the FDA back at that time, they wanted to have liver biopsy because at that time it was shown to be the best surrogate parameter for outcomes. I think the problem is with PSC that in order to study endpoints, those are very long-term studies, which probably take 5 to 6 years. And in terms of surrogate parameters, there's a lot of controversy. We know, unfortunately, that the non-invasive fibrosis test itself, FibroScan and also liver enzymes or ELF, is not reliable enough. In a way, I was surprised how well liver biopsy is performing in a heterogeneous patchy disease like in PSC, and discussing the slides with the pathologists, I think we are going to learn a lot about the pathogenesis of the disease. So in a way, I think we were lucky that we were forced to do liver biopsies, because I think it will be a breakthrough moment. Also, Sonia, it's a repository now to do those studies in a controlled, curated fashions with well-annotated liver biopsies. I think that will be a breakthrough, and as a community, we really have to work together to use this opportunity now.
Why don't we work our way down?
Yeah, so absolutely, I agree. The challenge in PSC research has also been the lack of a really good animal model. So this is where the biopsies become so important, and also just the rarity. So employing biopsies in a really open way where there's sharing across centers, I think will be a great step forward.
I'll take a unique perspective sitting on the patient side, but prior to this, I worked in the pharma side on 3 different PSC development programs, and have watched the challenges each step of the way, and in some ways it feels like we're actually stepping backwards at the regulatory level. So the fact that Michael and Norurso team actually succeeded in a phase 3 program is a huge milestone, and hopefully changes some of the thoughts from the regulators in an area where we don't have treatment. But something that I think is important for us as a community that has to get out there is the concept of shared risk. And the FDA says that they're a risk benefit, or they are a safety agency, and I respect them for that. But I think where we need to mobilize more is how do we bring that voice of we're willing to take some chances and some leaps of faith to get to where we may have a therapy. And it's going to have to be beyond biopsy and things. I think it's great we have that first step. But those, is it that you got lucky with the histology? Is it the drug? There's so many, we've had other studies where we thought the drugs worked, but the biopsy just didn't show it. So I think our voice and rallying that voice and working with pharma partners to make sure that they hear that we need a treatment and we want to do it safely, but I think patients are willing to take more chance than the agency is willing to actually give. So I encourage us to start to come together as a group and think about that.
I have about 4 pounds of insightful questions here, so we'll get to as many as we can. And there's been a couple questions about drug repurposing. What drug repurposing is, if there's a FDA-approved or European equivalent drug being used for a different reason, can it be repurposed for PSC? And the advantage is very much, a lot is known about the safety of the drug, the dosing of the drug. So the questions are, twofold. One, can any of the recently approved drugs for MASLD, fatty liver, or more generally other drugs, be repurposed for studies in PSC?
That's an interesting question. I mean, basically in MASLD, there's now resmetirom and the GLP-1 agonists. Starting with GLP-1 agonists such as semaglutide, an unknown fact is that bile ducts are the only cells in the liver which have a GLP-1 receptor. And we and others have shown that GLP-1 is protecting bile ducts from bile acid-induced apoptosis. So I could imagine, that there may be an added benefit, and maybe one is looking for signals in patients with comorbidity, or is looking what is happening to the bile ducts in patients with MASLD. There's a lot of going on with bile ducts as well. And resmetirom, which is a thyroid hormone receptor beta agonist, it also modifies bile composition in a positive way. I mean, having said that, both compounds may tweak it a little bit, but I don't think that will be a major breakthrough. Maybe, looking at PBC drugs, we've heard about elafibranor now with the positive phase 2 study, which I found very encouraging. The same applies to seladelpar, which is also a very interesting compound, which could be moved into PSC treatment as well. And I'm a big fan of repurposing. I mean, basically bezafibrate. I mean, this is how the entire story started. At the end, it's about safety. And when it comes to PPAR agonists, fibrates, bezafibrates, it's not that trivial as it would think. There are about 20% of patients experience side effects which lead to stopping the drug. And that's why I think we still need to improve that pathway.
So I'm also a big fan of drug repurposing, but more mechanism-based drug repurposing, so that we can kind of make sure that we know exactly what cells we think would respond to that drug. And we've had this conversation last night at dinner, again with Matthew and Andy, and then we've had this conversation with Steve. And one of the things that's interesting about the PPAR delta story is that it was tested in PSC, but not thoroughly tested in PSC. So this is where, in our lab, we're actually testing PPAR delta in the human slices, PSC slices, and the PSC organoids to try and understand, no one knows the mechanism of how PPAR delta works in PBC. And so if we can pull out some mechanism, and the concern is that these PPAR delta trials in PSC failed because of the wrong dose or the wrong way it's delivered. So I think the more kind of work we do to understand the mechanism of how those drugs work, the more likely we are to make it, or to be able to give good evidence, for preclinical evidence for a trial.
The hurdle we have to get over is, unfortunately, like repurposing, especially what's beneficial to the community is if these drugs are generic or they're readily available already and can be studied. The fact is that very few companies are willing to support that development, so it becomes up to us, up to academic collaborators. How do we actually take on, like what's been seen in other areas like HIV? Where do we as a community network start to say, okay, this is about driving and finding a therapy, And it's a fine line because you want to sponsor new therapies as well and make sure they get developed. But the pathway to get a repurposed drug studied appropriately is a challenge. But again, it comes, I think it's on us to actually have to think about how we do that.
And that's probably a space where really, programs like PSC Partners could help, because as you mentioned, industry won't have an interest. Yeah. And actually doing those kind of investigator-driven studies has become very complicated and expensive, at least in Europe, due to recent regulatory changes, which have made it really very, very, just to file the protocol, you already need 20,000 euro to file the protocol with the agencies. And I didn't mention one drug which is undergoing repurposing. That was an omission from my side. There's a big European study on statins in Scandinavia, which actually look at endpoints. So it's really a 6 or 7 year study, and they look at clinical endpoints. So that interesting, statins have a very pleiotropic effect. That will be very exciting to see what the outcome of that study will be.
This question, the answers may be a little speculative, but there's several questions about AI. If any, what would the role of AI be in both designing drugs that may be effective and perhaps digitally modeling the use of these drugs prior to clinical trials?
Yeah, at that point, AI was not yet used because all those study designs were, performed before the hype of AI. Personally, I think perhaps not so much the design, but the data analysis AI will play an enormously big role because, and maybe Sonia will comment on that, the complexity of data which we are gathering there, the molecular, metabolomic, clinical, radiomic is enormous. So I think the only solution for that will be AI. And actually in the programs which we plan to set up to analyze, for example, all the future data from this urocolic acid data, there's a big AI component in that. In terms of drug design, I'm a little bit hesitant. In the future, perhaps. At the moment, not yet.
Yeah, it's an excellent question. It's something we talk about a lot in the Human Cell Atlas program. And one of the big points is that the data going in has to be very well informed. So, for example, in our single cell data, we generate like huge amounts of data, and it can lead to 300 hits. But if we don't kind of match that with what our pathologist is seeing in the cells, so they say there's this random, T cell that is completely different in PSC. We should target that. But then the pathologist says there's about 1 or 2 in the whole biopsy. So what we need to do is kind of have AI do its thing, but then make sure that we're always informing. And so that's why we're seeing mechanism-based drug repurposing. We say, we know from the pathologist that there's a ton of macrophages around those lesions, within the lesions, and they look like this, and there's other macrophages that are outside the lesions in the healthy area that are chugging away, promoting regeneration. So this is something that AI will not know. So we'll say, only focus on these populations, and now tell me what drug is possible that could possibly be used. And that's how we came up with, on Thursday, the SYK inhibitor. The AI algorithm said, you can use SYK to target the macrophages within those lesions, and actually SYK is being employed in another inflammatory disease. So AI is definitely going to be part of the tool, but it also has to all be driven by the clinical eye.
I see AI, I think everybody's jumping to AI being, the pinnacle of everything, and I think for me it really is an efficiency tool, and I think when you look at clinical development and drug development, there are plenty of places where AI can actually help us on the industry side or in the research side just move things faster. I think what we hear consistently from everybody is, how can we move things along faster? And so I always think of AI as, can it save us time in doing something that we've been doing manually? Can it help us identify networks of patients who don't have access to trials? Can we cull data in different ways that can just speed the process along? Maybe not take us to this hyper, I think some of the work you guys are doing is great, but I think even just the basics of how do we move things faster, it can have some very simple basic benefits.
There's several questions about percutaneous liver biopsy or transjugular liver biopsies. 20 years ago, liver biopsies were very frequently done, and for a whole host of reasons. And the holy grail was to develop technologies that we would never need to do one. And this is true whether you're a cardiologist in the heart, et cetera. But I think the role of non-invasive assessment of fibrosis versus the need for liver biopsy is pretty unique when you're addressing the question of research. So perhaps the three of you can comment on the role of liver biopsy being required versus alternative mechanisms in clinical or basic studies.
Right, yeah. I mean, for example, in Europe or at my center, we still do the liver biopsies as hepatologists ourselves. I think it's a very important part of this trustful patient-doctor relationship. In my experience, I never had problems, for example, in convincing my patients where there was a long-lasting relationship also to undergo a liver biopsy. I think, and of course it's important to make it safe, to make it painless. You can do that also under sedation or with appropriate medication. And at the end, of course, what I think, liver biopsy may not be the ultimate way to go in PSC because it's such a patchy disease, right? So in a way, I was surprised that in our study we got this clear result. At the end, maybe what we are also trying to do is fusion or guided biopsy, for example, like is done in other organs, for example, prostate. There would be fusion biopsies where we really target a specific affected area. The same could be done in liver. Maybe this is the way to do. Also with minimal invasive endohepatology, with endoscopic ultrasound, so that you no longer do that percutaneously. And actually, because you've mentioned transjugular liver biopsy, we have a very huge hemodynamic program in Vienna with more than 350 procedures a year. And sometimes we offer patients, do you want to have a transjugular liver biopsy instead of a percutaneous liver biopsy? And the preference is for transjugular because just simply psychologically it seems to be less, repelling. I think that endoscopically guided biopsies from the stomach, where you also can guide maybe the direction based on MRI fusion images like we do it in other organs, that may be the way to go. You also could non-invasively or minimally invasively access the portal vein or also the bile duct itself, because you don't cause the pancreatitis like you do when you do it from the gut. So I think what we call endohepatology is an entire new discipline of doing endoscopic procedures, endoscopic ultrasound guided by hepatologists. But the challenge is to train people doing that. So this is extremely complex. It takes time also for doctors to reach this skill. And sometimes I have the impression we avoid of developing certain techniques because just simply we think it's too resource demanding. I don't know what patients would think about an endoscopic guided, ultrasound guided liver biopsy. You could imagine maybe even as a one-stop shopping in the same setting, like you screen for varices, you could do the whole nasty stuff in one session basically, right?
We, as an organization a few years ago, we took somewhat of a stand against liver biopsy as being an endpoint because to that date it had really failed. And for all the reasons you brought up. And so we really were, pushing for other non-invasives. We were definitely behind, where MASH was. MASH is just getting to where like FibroScan might be able to make it there in a few years, and we may be able to kind of link to that. But I think one thing that the NORURSO trial has done, and I credit both Falk and frankly your leadership on this, is actually working very closely with the regulators on how do you look at the biopsy. I think it's been so shotgun in the past that it just had, the scoring systems haven't been well defined. Working to make sure that if you're going to get a biopsy, it's telling you the best signal it can. And I do think the trial, that you helped lead is really may change the thought on that, but it was a partnership to make sure that histology and the reading of that was done in a way. So then patients feel like if I'm going to undergo it, that there's a really good chance it's going to mean something in a trial. And I think that's a huge piece of it.
I mean, maybe to close the biopsy chapter, I was also in PSC, I was very much against liver biopsy, I must say. Now I've changed my attitude because we got so much information out of it. Just let me tell you, in nearly 10% of the patients were not allowed to enter the trial because something else came out as the diagnosis. Sarcoidosis, you know? I mean, in centers who really know how to diagnose PSC, you would be totally surprised. Mast cell cholangiopathy. I mean, and those were people who really knew their patients and know how to diagnose PSC. So this just simply shows how heterogeneous the disease is, and just simply the cholangiographic feature itself is telling us not enough. That's my opinion.
I also think that the role in a clinical trial where you can do a non-invasive monitor may be distinctly different from if you're doing a basic investigation, how the immune system is interacting with various parts of the liver.
But in the basic investigations, one of the things, I mean, it's absolutely extremely valuable to have the biopsies, but one of the things that you have to do is, as opposed to saying, here's 8 biopsies, you say, actually, here's 30 portal to central venous areas with this score for each portal to central venous areas. So you're correlating everything with the histological features as opposed to saying, these are all PSC biopsies. And then you pull out, here are the regenerative signals at this stage of fibrosis or this degree of fibrosis. So it's having the pathologist take each biopsy as an ecosystem, not as a fixed kind of diagnosis.
Okay, Michael, you have 30 seconds for this one. What's the difference of urso and nor-urso?
The difference between what?
Urso and nor-urso.
It's a chopped off version of urso, which makes it resistant to conjugation, you know. So basically, conjugation is a way to keep it within the circulation between the gut and the liver. And by modifying that, you keep it between the bile duct and the hepatocytes within the liver. So you basically enrich it to the bile duct, which is the site of action.
But is it safe to say they sound the same, but they work completely different? They're different drugs.
That's why it was also renamed, because people, you know, always mixed it up with ursodeoxycholic acid. So norocolic acid, I hope there's no mix-up with obeticholic acid, right?
We heard yesterday, and we've heard about what are called IBAT inhibitors. When you secrete bile acids, it goes in the intestine and it's reabsorbed in the ileum, which is the small intestine. And 95% of your bile acids are not being made, they're just recirculating. So in the past couple of years, there's been 2 FDA drugs that block the reuptake of it. And we heard a little about it yesterday, but what would the role of potentially IBAT inhibitors be with PSC, both alone or even more important, in combination with other therapies?
Is that also directed to me? I don't want to have a
We'll work
monologue,
our way
right?
down.
We can go right to left, boy, girl, boy, girl, I don't know.
I think IBAT inhibitors are more symptom-oriented pruritus, but in preclinical mouse models, like in this mouse model which I showed you, IBAT inhibitors also reverse cholangitis by making the bile less toxic. So let's see, maybe it's doing more than just relieving symptoms, especially when combined with other drugs. So at the end, I think we'll have 2 or 3 drugs which we'll combine.
I think the unique aspect for PSC for IBATs, and they obviously are working in other cholestatic diseases. I'd be very surprised if we don't see positive results in a PSC study, but we've all been shocked. I think it gets a drug approved because you can get approved on a way a patient feels, functions, or survives. It's the quickest way to get a drug approved right now is based on a symptom. But what it allows us to do is get something that's an important symptom to be treated, but then patients can be followed with a drug out there for a longer period to see what are the next levels of benefits. I mean, I'd be willing to bet we're going to see some improvements in fatigue. You know, you're getting some subtle signals, and in other more pediatric liver diseases, there may be outcomes improvements. But getting something available, having a win of a drug that can be approved relatively quickly, it builds the momentum of getting therapies out there. So fingers crossed, that may be our one, at least in the U.S., maybe one of our first benefits.
Many questions about the role of impact of federal funding changes in the U.S. that probably are not answerable at this point. But I think one thing that may be relevant, perhaps Steve, you can start on it, is what we can do as a community with donations and fundraising and pilot feasibility studies for PSC. How can we help this problem?
Yeah, I mean, it's interesting now as we go to the International PSC Study Group and we're announcing, you know, wind was launched. Wind is going to end up being close to a $20 million project by the time we finish. It was launched with $3 to $4 million of just patient funds. So the patients are behind it, and the patients can become a very powerful driver in moving these things. The community is now being looked to, like with our grants program, more. It's why we have 40 applications now, because we are starting to be able to fund, you know, at least starting people out. It's going to be even more critical now how we do these things, because, and there's no two ways about it, funding is not going to get better in the next year. So how we, and it's not just individual donations, how do we go out and find other avenues for funding as a community? It doesn't even have to be you as an individual. How can we network to look at other sources we haven't thought about? Other corporations outside of pharma, things that can really help move and make our disease and our story is so compelling when it gets in front of a variety of people, not just people in the healthcare industry. And how we put that together is really key, and I think it's going to be important.
Yeah, I think one thing, one last comment, and then we'll draw this to a close. As we're asking for funding, or even if we're not, it's incumbent on us as researchers to let you know what we're doing and why we're doing it and how we think this will help, because you're the people who are either having this disease or closely related. And if we're going to perform research, we must show all of you why we're doing it and the early or long-term rewards of it. And I want to thank the three investigators here for what I think was a truly outstanding way to start the day today.
Okay, very good. Yeah, that was exciting discussion. Nicely. Okay.